Hello students, I'm sorry that I could not send a video in these 2 days, and today we are going to discuss a new topic, a new paragraph which is given below the paragraph of lipids. Okay, so in the last video we have learned about lipids. Before that we have covered nucleic acids, and today we are going to learn about nucleosides and nucleotides. This paragraph is explained after the paragraph of lipids. Okay, last time we have learned regarding lipids, and today we will learn about nucleosides and nucleotides. So these nucleosides and nucleotides are a kind of carbonic compounds which are having heterocyclic rings in their structure. Okay, nucleosides and nucleotides are a kind of carbonic compound. They are a type of carbonic compounds in which heterocyclic ring is found, and these carbonic compounds are found in the acid soluble fraction when we do the chemical analysis on the tissue. Okay, when we perform the chemical analysis on the tissue, the nucleosides and nucleotides are found in the acid soluble fraction and so their molecular weight is less. So molecular weight of nucleoside and nucleotide is very less, and they are having heterocyclic ring in their structure. Nucleosides and nucleotides have heterocyclic rings in their structure. So now what is this heterocyclic ring? We will talk about that first, and then we will discuss nucleotide and nucleoside. We will talk about the heterocyclic ring which is present in the structure of the nucleoside and nucleotide, and then we will discuss the structure of the nucleoside and nucleotide. So let's talk about heterocyclic ring. So the heterocyclic ring which is found in the structure of the nucleotide and nucleoside is known as nitrogen bases. Nitrogen base. So the heterocyclic ring that is present in the nucleosides and nucleotide is known as nitrogen base. And how many types of nitrogen bases are there? There are two types of nitrogen bases that are found in nucleosides and nucleotides. One is known as purine type of nitrogen bases, and second is known as pyrimidine type of nitrogen bases. Two types of nitrogen bases are there: purine type of nitrogen base and pyrimidine type of nitrogen base. Both these nitrogen bases are found in nucleotides and nucleosides. Purine nitrogen bases are having two rings. They are having two heterocyclic rings in their structure, whereas pyrimidine nitrogen base has only one heterocyclic ring. It is having only one heterocyclic ring in the structure, so they are known as pyrimidine. Okay, so what is nitrogen base? Nitrogen base is a heterocyclic ring which is found in nucleotide and nucleoside. How many types of nitrogen bases are there? Nitrogen bases are of two types: purine nitrogen base and pyrimidine nitrogen base. What is purine nitrogen base? Purine nitrogen base is that nitrogen base which is having two heterocyclic ring in their structure, whereas pyrimidine nitrogen base is that nitrogen base which is having one heterocyclic ring in their structure. Okay, so let's draw the figure of purine and pyrimidine. So if this kind of figure is there, this is one heterocyclic ring, and along with that this kind of heterocyclic ring is found. This is one heterocyclic ring, this is first heterocyclic ring, and this is second heterocyclic ring. And on the top of this ring, if amino group is there, then this nitrogen base is known as adenine. It is known as adenine type of nitrogen base. One heterocyclic ring, second heterocyclic ring. Okay, this is hexagon type and this is pentagon type. Okay, this is hexagon type and this is pentagon type. So two heterocyclic rings are there, and on the top of this ring amino group is present. So this nitrogen base is known as adenine nitrogen base. Okay, now let's draw another structure. This is a hexagon heterocyclic ring, and along with that a pentagonal heterocyclic ring is also there. Okay, so This is a hexagonal ring, this is a pentagonal ring, and on the top of this ring oxygen is present. On the top of this ring, what is there? Oxygen is there. Then this nitrogen base is known as guanine. It is known as guanine. This is known as guanine. So these two are known as purine. These two are known as purines. Adenine and guanine are known as purines. Okay. On the adenine, two heterocyclic rings are there. One is hexagonal, one is pentagonal, and on the top of the ring, amino group is there. In the guanine, the two heterocyclic rings are there, and on the one is hexagonal, one is pentagonal, and on the top of the hexagonal ring, oxygen is present. So it is known as guanine. So two nitrogen bases are known as purine. They are adenine and guanine. Let's talk about pyrimidine nitrogen bases. There are three examples in pyrimidine nitrogen bases: cytosine, thymine, and uracil. Cytosine, thymine, and uracil. So let's draw the cytosine first. Here only one single ring is there. Here one and two rings were there. One and two rings were there. Here only one heterocyclic ring is there. And on the top of this heterocyclic ring, amino group is present. On the top of this ring, amino group is present. Then this nitrogen base is known as cytosine. It is known as cytosine. Cytosine. This is known as cytosine nitrogen base. Okay. Let's draw the uracil. Uracil. One heterocyclic ring is there. On the top of this, oxygen is present. On the top of the ring, oxygen is there. Then this nitrogen base is known as uracil. This is known as uracil. Uracil. Is it clear to you? One heterocyclic ring. On the top of that, amino group is there. Then it is known as cytosine. One heterocyclic ring. On the top of that, oxygen is there. Then it is known as uracil. Okay. Then one heterocyclic ring. On the top of that, oxygen is present, and on fifth carbon, if methyl group is present, on the fifth carbon, what is there? Methyl group is there. See, here fifth carbon is there. This is first carbon, second carbon, third carbon, fourth carbon, and fifth carbon. On the fifth carbon, if methyl group is present, then this ring is known as thymine. It is known as thymine. Okay. So there is not much difference between uracil and thymine. On uracil, only oxygen is present on the top, whereas in case of thymine, the methyl group is present at the fifth carbon position. Okay, so thymine is also known as 5-methyl uracil. Thymine is also known as 5-methyl uracil. Okay, another name of thymine is what? Another name of thymine is 5-methyl uracil. So what we are talking about nucleosides and nucleotides, and we said that nucleosides and nucleotides are a kind of carbonic compounds which are having heterocyclic rings in their structure. Okay, and nucleosides and nucleotides are found in the acid soluble fraction when we perform the chemical analysis on the tissue. Okay. And then we said that before discussing about nucleotides and nucleosides, we have to talk about the nitrogen base. What is nitrogen base? Nitrogen base is a heterocyclic ring which is found in nucleosides and nucleotides. Okay, and these heterocyclic rings can be are of two types. It can be purine or it can be pyrimidine. Okay, purine have two heterocyclic rings in their compound. Pyrimidines have only one heterocyclic ring in their compound. Okay. Then purines are known. There are two examples of purine, adenine and guanine, and there are three examples of pyrimidine: cytosine, uracil, and thymine. Here two rings are there. One is hexagonal ring, one is pentagonal ring. Pentagon is attached to hexagon, and on the top of that amino group is there. Then that nitrogen base is known as adenine. Here hexagon is there, here pentagon is there, and the top of the hexagon oxygen is there. Then it is known as guanine. So these two are known as purines. Purines are made up of two rings. And examples of purine are adenine and guanine. Then pyrimidines have one ring in their structure. Pyrimidines are made up of only one heterocyclic ring. So here one heterocyclic ring is there, and on the top of that amino group is there. Then that structure is known as cytosine. Here one ring is there, one heterocyclic ring is present, and on the top of that oxygen is present. Then it is known as uracil. And one heterocyclic ring, and on the fifth carbon position methyl group is present, then it is known as 5-methyl uracil or thymine. Okay, so here there is not much difference between uracil and thymine. Okay, so these are known as nitrogen bases. Now this nitrogen base is attached. It is attached to pentose sugar. Which kind of sugar? Pentose sugar. When the nitrogen base gets attached to a pentose sugar, then the resultant structure is known as nucleoside. When a nitrogen base is attached to a pentose sugar, then a resultant structure is known as nucleoside. Are you getting this? These are nitrogen bases. Okay. Out of this, any nitrogen base can get attached to a pentose sugar. Okay. Out of this, any nitrogen base. It can be thymine, it can be uracil, it can be cytosine, it can be guanine, it can be adenine. Any nitrogen base can get attached to a pentose sugar. Now what is pentose sugar? A sugar which is having 5 carbon is known as pentose sugar. Okay, sugar which is having 5 carbon is known as pentose sugar. So when a nitrogen base which is attached to a pentose sugar, the resultant structure is known as nucleoside. Okay, and if an additional phosphate is added to the nucleoside, okay, if extra phosphate is attached, okay, if we attach a phosphate group to the nucleoside, okay, if we attach a phosphate group that is in form of phosphoric acid, okay, phosphate means phosphoric acid. So when we join nucleoside with a phosphoric acid, or when we attach a phosphoric acid to a nucleoside, then the resultant structure is known as nucleotide. It is known as nucleotide. So I hope you are getting this very clear. Okay, when we have learned regarding the nitrogen base first, okay, and then we said that total 5 type of nitrogen bases are there, okay, adenine, guanine, cytosine, thymine, uracil. Okay, out of this, any nitrogen base can get attached to a pentose sugar which is having 5 carbon in their structure, and they will form nucleoside. Okay, so what is nucleoside? Nucleoside is sum of pentose sugar plus nitrogen base. Nucleoside is sum of pentose sugar plus nitrogen base. When we join extra phosphoric acid or phosphate group to this nucleoside, when we join extra or additional phosphate or phosphoric acid to the nucleoside, then the resultant structure is known as nucleotide. It is known as nucleotide. So I hope you are clear with this. A nucleotide is having 3 components. Nucleotide is having 3 components: nitrogen base, pentose sugar, and phosphate. Okay, whereas a nucleoside is having only 2 components: nitrogen base and pentose sugar. Nitrogen base and pentose sugar. Now let's draw the structure of the nucleoside and nucleotide. I hope you are clear with this nitrogen base and the definition of the nucleoside and nucleotide. Now we will draw the structure of the nucleoside and nucleotide. Let's draw that. Sure. Say for example, I am going to draw a pentagon, and that pentagon represents a pentose sugar. Okay, I am going to draw a pentagon, and that pentagon represents a pentose sugar having 5 carbon in it. Okay, so this is a pentagon. This is a pentagon. This is a pentagon, and this pentagon is having 5 carbon in it. Okay, here on this position oxygen is present. On this is known as first carbon, this is known as second carbon, this is known as third carbon, this is known as fourth carbon, and out of the ring, this is known as pentagonal ring, and out of the ring an extra carbon is present in form of CH2OH. Extra carbon is present in form of CH2OH, that is known as fifth carbon. That is known as fifth carbon. Okay, so here a pentose sugar is there, and the counting of the carbon starts from here, not from here, because here oxygen is there. Here carbon is located, so counting of the carbon will start from here, Here first carbon is there. Okay, I have not drawn the carbon, but I am indicating that carbon is present over here. Okay, so here first carbon is there, second carbon is there, third carbon is there, fourth carbon is there, and fifth carbon is present outside of the ring. Fifth carbon is present outside of the ring. Okay, now say for example, on the first position, a nitrogen base is attached. On the first position, a nitrogen base is attached. Okay, this is not an actual figure of the nitrogen base. This is just a representative structure. Okay, so this is a nitrogen base. Okay, I have drawn it like this. This is a representative structure, not original one. Okay, so this is a nitrogen base which is added to the first carbon of the pentose sugar. This is a nitrogen base added to the first carbon of the pentose sugar. Okay, and here a bond is there. Here a bond is present between nitrogen base and pentose sugar. A bond is present between nitrogen base and pentose sugar. That bond is known as N-glycosidic bond. N-glycosidic bond. That bond is known as N-glycosidic bond. Okay, and say for example, we have joined adenine over here. Okay, so now we have attached adenine nitrogen base to the first carbon position of this pentose sugar. So this structure is known as nucleoside. This is known as nucleoside. Okay, what is nucleoside? Nucleoside is a structure having pentose sugar and a nitrogen base. And which kind of bond is present between pentose sugar and nitrogen base? And glycosidic bond is present between pentose sugar and nitrogen base. Okay, so now let us add phosphoric acid on the fifth position of the nucleoside. Okay, let us attach phosphoric acid to the fifth position of the nucleoside. Now this is nucleoside. Okay, and now we will add a phosphate group. I am just drawing P. Okay, it is HPO4. Okay, so I am drawing only P. Actually it is HPO4. So we are adding HPO4 on the fifth position of this nucleoside. So this whole structure is known as nucleotide. This structure is known as nucleoside. Initially this much was there, which was known as nucleoside. Initially this much structure was there, which was known as nucleoside, but when we add an extra phosphate to the fifth position of the pentose sugar, then it is known as nucleotide. Okay, so what is nucleoside? Nucleoside is equal to pentose sugar plus nitrogen base. Now I am not writing whole nitrogen, I have written N base. Okay, so a pentose sugar is there and an N base is there. Okay, pentose sugar and N base. So this structure is known as nucleoside. And if we join extra phosphate to this structure, then the resultant structure is known as nucleotide. Are you getting this? So what is nucleoside? Nucleoside is a nitrogen base along with the pentose sugar. What is nucleotide? Nucleotide is a nitrogen base along with pentose sugar and the phosphate which is added to the fifth carbon of the pentose sugar. So that is known as nucleotide. Okay, so I hope you are clear with the structure of the nucleoside and nucleotides. Now let's see the examples of nucleosides and nucleotides. Okay, we will learn about the examples of nucleoside and nucleotide. Let's learn the examples. Okay, examples of nucleosides and nucleotides. Now I will initially give you examples of nucleoside first. Okay, I will give you examples of nucleoside first. So we will talk about nucleoside first. Say for example, a pentose sugar is there, and here adenine is attached. Okay, here adenine is attached. Then that nucleoside is known as adenosine. It is known as adenosine. Okay, here adenine is there and a pentose sugar is there, then it is known as adenosine. Okay, sure. Then if I draw thymine over here, and pentose sugar is there, then it is known as thymidine. Okay, these are known as examples of nucleosides. Adenosine is a nucleoside, thymidine is a nucleoside. Then if cytosine is attached, cytosine is attached to the pentose sugar, then it is known as cytidine. Cytidine. If guanine is attached, okay, then it is known as guanidine. Guanidine. Okay. And if uracil is attached, then it is known as uridine. It is known as uridine. Okay. So these are known as examples of nucleosides. Okay. Adenosine, thymidine, cytidine, guanidine, uridine. Okay. These are known. Sorry, guanidine, guanosine. Sorry for this. Guanosine, not guanidine. Okay. So adenosine, guanosine, thymidine, cytidine, uridine. These are known as examples of nucleoside. Please make correction over here. Okay. Initially by mistake I have said that it is guanidine, not guanidine, guanosine. Okay. So adenosine, thymidine, cytidine, guanosine and uridine. These are known as nucleosides. Now let's give example of nucleotides. Okay, we will give examples of nucleotides. So you all know that on the fifth carbon position, phosphate will be there. On the fifth carbon position, phosphate will be there. So it is known as nucleotide. And say for example, here adenine is there. Okay, so for example, adenine is there. Then this is known as nucleotide of adenine. It is known as nucleotide of adenine. And here phosphate is present in form of phosphoric acid. So this structure is acidic in nature. So this is known as adenylic acid. This is known as adenylic acid. What is adenylic acid? Adenylic acid is a nucleotide. Adenylic acid is what? It is a nucleotide. We are giving examples of nucleotides now. Initially we gave examples of nucleosides. Now we are giving examples of nucleotides. So adenylic acid is a nucleotide. Okay. Now let's talk about this thymine. Then it is known as thymidilic acid. Thymidilic acid. Okay. Then it is known as thymidilic acid. If guanine is there, then it is known as guanilic acid. Guanilic acid. Okay. If cytosine is there, then cytidilic acid. Okay. And if uracil is there, then it is known as uridilic acid. Okay. So these are known as nucleotides. Okay. Adenylic acid, thymidilic acid, cytidilic acid, guanilic acid, uridilic acid. Here acid is there because phosphoric acid is present on the fifth carbon of the pentose sugar. Okay. Phosphoric acid is present on the fifth carbon of the pentose sugar. So this is all about nucleoside and nucleotides. These are examples of nucleosides and these are examples of nucleotides. Please remember, I have just explained you one paragraph which is given below the paragraph of lipid. Okay. In the next video, we will talk about primary and secondary metabolites. Okay. So till then, please learn this video well, and we will meet in the next video, and then we will talk about primary and secondary metabolite. Okay. Chalo, best of luck.